Welcome back to the NWR Virtual Healthcare Conference. Next up, we have Dr. Michael Baker, Managing Director and CEO of Arovella Therapeutics. Arovella is a biotechnology company focused on its invariant natural killer T-cell therapy platform, or iNKT, from Imperial College London, and this is to treat blood cancers and solid tumours. Arovella is also expanding into solid tumour treatment through its Claudin 18.2 targeting technology that it licensed from Sparks Group. And it's also working towards additional tumour targeting technologies that are anticipated to be used in conjunction with the iNKT cell therapy platform. Just a reminder, any questions, please feel free to type them in using the Q&A panel within Zoom. But I'll now hand it over to Michael.
Right. Thanks, Matt, very much for the introduction, and thank you for putting the event on. And for everyone that's out there listening, thank you very much for taking the time. I'm looking forward to giving those that know the company an update on our programs, and of course, those of you that are new to it, I look forward to taking you through the story at Arovella Therapeutics. So please do note our disclaimer. I will be making forward-looking statements. And just starting with the highlights of the company. So what we're working on, as Matt alluded to a moment ago, is its invariant natural killer T, or iNKT cell platform for short. And in brief, we're using that to target both blood cancers and solid tumours. Now, our lead product is called ALA-101, and that is being developed for what we call CD19 positive blood cancers, so little cancer cells that have CD19 on their surface, and we're anticipating to take that into phase 1 clinical trials, which we're very excited about. Now, the cell therapy sector, it has transformed the way we think about cancer treatment, and that's great, but we know there are also some challenges that have arisen in CAR-T's current format. And so we believe that it's our iNKT cell therapy that is well positioned to solve some of those challenges in the sector. Now, in terms of our leadership, I won't go through it today, but I think we've assembled a magnificent team that's both at board level and management level that has a lot of expertise right the way through from R&D into drug development, but also into commercial transactions as well. So in terms of our model, we have largely built out of a strategic acquisition approach where we license what we consider to be the best IP globally, and that's how we started work on our iNKT cell platform. We've continued to build out through very careful strategic licenses since that time, and we continue to survey the landscape for things that we think enhance our platform specifically. So for those reasons, in addition to the fact that we are one of a handful of companies globally working on iNKT cells and the only company listed or otherwise in Australia working on an iNKT cell platform, we do see that we are positioned well for growth. So just a little bit more on our IP strategy broadly. So the way we've worked, as I said, we are very careful and selective about the IP that we bring in. So we don't have a scattergun approach and many different technologies that we need to get our heads around. We have our foundation IP from Imperial College London. We have an armoring technology from the University of North Carolina, which we're implementing for our solid tumour programs. And we've also just licensed recently a novel, what we call CAR, from Sparks Group, which gives us shots on goal to target things like gastric cancer and pancreatic cancer. So building out either the performance and the utility of the platform. And then we overlay that with good regulatory strategy where we consider pathways available to us, which may help us to either accelerate approval or to essentially enjoy market exclusivity at the right time. And then the last pillar of our IP really is around know-how, and this is quite important for things like cell therapies where there's an enormous amount of know-how that goes into the manufacturing of these types of therapeutics. Not to dwell on it here, but these are just two examples of our patents that we've licensed from, on the left, Imperial College London, on the right, Sparks Group, both with granted patents in certain territories, as well as filings underway in most major territories as well. So in terms of the company's financials, I think you can see here from the share price on the right, we've had significant shareholder growth over the last 12 to 18 months, which has been excellent. And we think that is off the back, again, of some data we presented at the American Association for Cancer Research last year, as well as the strategic license agreements that we've put in place over the last few months or so. So certainly over the course of the next 12 months and beyond, we're looking forward to continuing to hit the milestones that we've set in place and hopefully delivering additional shareholder value over the months and years to come. Now, in terms of our cash balance, we're currently, we finished last year just under $5 million cash in the bank. So we're in a good position from a capital perspective, and delighted with our shareholder base, where our largest shareholder Merchant has been very supportive, as well as a number of high net worths and smaller institutions that all have also continued to be very supportive of our activities. Now, I think it's important to highlight this, just how important the cell therapy sector has been over the last 2 years, and that's indicated by the transactions that have continued to transpire in the space. So many of these bar one early stage phase 1, phase 1B clinical trials, except for that phase 2 one there, stage companies. And what we've continued to see is a range of either licenses, acquisitions, or strategic partnerships where the upfronts are often in the range of $200 million or thereabouts. And for some where they look a little bit lower, there's often an equity investment or something else tied, which also builds those up into the $200 million range. And worth noting, AstraZeneca acquired Gracell at the end of last year, and that was finalised only a few weeks ago, and that was $1 billion up front. So the major takeaway from this slide is that we agree here that the cell therapy sector is still something of immense value to large pharmaceutical companies, and again, delighted that we're working in that space directly. So in terms of CAR-T, a little bit of a refresher here. So what exactly is it? And I think a good way to think about CAR-T specifically, it is the ultimate personalised medicine. I'll explain why. So when we think about a patient that may have a lymphoma leukemia, they will have failed certain treatments beforehand. And then what actually happens is their blood is taken, and we actually collect the immune cells from their blood called T cells. So they consider those as like the soldiers of the immune system, which essentially survey the landscape for things that don't belong, like microbials or even cancer cells. But in the face of cancer, they become overwhelmed. And so we end up in a situation where we need to enhance the immune cells. So we take them to a lab. They get genetically reprogrammed to have a CAR on the surface, the little red and blue claw there. And you can consider those as almost like homing missiles that essentially direct the immune cells to the particular form of cancer. They end up getting grown up in a lab. They'll be infused back into that patient. And then anywhere, in this example, the little green dots on the cancer cell, anywhere they're located, the immune cells will now find them and destroy those particular cancer cells. So a big important point here is that we start with the patient and then we go back to that same patient for manufacturing. Now, notwithstanding what we've seen over the last few years or so is that it has been remarkably successful in blood cancers. We are now starting to use the word cure and cancer in the same sentence, which is phenomenal. Not surprising for the first 2 products that were approved initially. Last year in 2023, we saw combined sales of more than 2 billion. So we're very comfortable here that this is a pillar of oncology that's here to stay. And this is just one example here, but an example showing that by 2030, this will be a market worth more than $60 billion. Now, important to point out, like all cancer therapeutics, this is not curative for every single patient. And we do still see that in the order of 40 to 60% of patients do relapse post CAR-T therapy. I like this example here. It's a good example of a young lady called Emily Whitehead. She was 7 when she took this type of therapy experimentally, now 12 years ago. So just to be clear, this is the same girl over 10 years, but now she still continues to be 12 years on, 12 years cancer-free, which is excellent. Now, I referred to it a moment ago, and unfortunately for all these things, there are some corollaries and challenges. And the big one here for CAR-T is that concept that it starts from the patient and finishes with the patient. So what we know out of that is it makes the therapy very expensive from a cost point of view. So it's about $500,000 US per dose. And because we're starting with the patients that actually have cancer, it's not unsurprising that their immune cells are compromised because they have seen cancer, they've seen chemotherapy and other treatments. Not all treatment centers can actually make and manufacture these types of therapies. So there may be patients that could be eligible to take it that would miss out. And because it's being made bespoke, it actually takes about 3 to 4 weeks. In some cases, it would be longer for patients to actually get the therapy. And we do see that patients can succumb to the disease within that time frame. And of course, because it's being made every time bespoke, it does mean that manufacturing run failures can and they do occur. So what we're proposing at Arovella is that we work with our iNKT cell platform and we create or develop an allogeneic or off-the-shelf cell therapy. And what that means is that we're actually collecting the immune cells from a healthy donor. We manufacture those iNKT cells to produce the CAR, and then we're able to give those to many patients after they've been manufactured and stored in the freezer. So the idea here is that this actually then could end up in a patient less than 1 week, let's say, for them to actually receive a particular dose once they've been shown to be eligible to take the therapy. So what is it about iNKT cells exactly that enables us to do this? And let me just not go into an immunology lesson here, but just to give you an overview of why this is the case. So we have T cells on the right here in blue, and natural killer cells, which has also been the focus of a range of other cell therapy companies. But as I said, what we're most interested in is the iNKT cells that sit in the middle. And a big factor here for why we can use iNKT cells is the fact that they don't cause graft versus host disease. So what happens with T cells is they will go in if they went from a healthy donor to a patient, they would recognize things in that patient as foreign, and they can start attacking tissues, organs, and so forth. And that can be fatal in the form of graft versus host disease. So we don't want to do that. So iNKT cells, on the other hand, they've been shown in clinic that they can be given from one person to another patient, and there is no graft versus host disease. So we already know that we can give these from one person to many people. Now, there are other benefits of iNKT cells I'd like to point out. There's 2 circles here for a reason. One is our innate immunity, which is our fast-acting immune response. And the second is our adaptive immunity, which is a more long-term durable response. And iNKT cells are capable of both. Now, they're often considered the front line of the human immune system, and that's where we get the very fast-acting component from. And they actually incorporate killing mechanisms from both T cells and natural killer cells, so they can do both killing jobs. And I'll talk more about it as we get into it, but they also have multiple properties that we think make them quite suitable for use in tackling solid tumors. So it's important, again, just thinking about the landscape here. We look at the T cell on the left and the NK cell sectors on the right. And what we know is there's hundreds of companies, close to thousands of clinical trials ongoing for both those two combined. So for Arovella, where we sit today, it's in the iNKT cell space. And as I mentioned a moment ago, there is still a handful of companies working in this particular sector. So we do see that we're in a wonderful part of the cell therapy landscape, and certainly in a differentiated position. And given that we've continued to get new cars and to get new armoring technologies, we're further differentiating ourselves also within the iNKT cell setting as well. So in terms of how we intend to manufacture, as I said, what we'll be doing is collecting healthy blood from a donor. We then are able to isolate their iNKT cells. Once we've isolated the iNKT cells, same steps apply. We genetically reprogram so that ultimately they have a CAR on their surface. As I said, think about that like a guided missile. They get expanded or grown up into the billions before ultimately being put in small vials, frozen, and then stored, ready for use. When a patient's eligible to take the therapy, we can then take that out, thaw it out, and it's able to be given to those patients. So some key advantages here are, because we're not working with the patient, we believe that the immune cells are in better shape initially, so we have healthier starting material. We're able to make multiple doses per batch, so it's scalable. We can get faster access to treatment because we're not having to manufacture just in time for every patient. And once they're in the freezer, we've essentially mitigated the risk of any manufacturing run failures. So I would like to show some data on the iNKT cell programs. I think it's always helpful to show why we're also so excited about this from an efficacy point of view. So this is a tumor model that expresses 2 targets that our lead product ALA1 will recognize, CD1D and also CD19. And what we do in these studies is we actually infect or engraft the cancer cells into the animals. So anywhere you see the color, red being the most intense, that's actually cancer cells in the animals. So day 0 is the top 2 rows, day 3 is on the bottom 2 rows. And in the first column, we treat with phosphate buffered saline. Middle, it's T cells without any guided missiles or armoring or engineering, I should say. And then iNKT cells also without any engineering or CAR-T or CAR, I should say. And then here in orange, it's the closest comparator we could have to something that Emily Whitehead would have taken, which is a CD19 targeting CAR-T. And the last column is ALA101. So what we're looking to see on the bottom 2 rows actually is elimination of that color or lack of color, which indicates lack of tumor cells. So we're very comfortable here on the bottom right that the activity from the iNKT cells, and specifically within 3 days, is incredibly quick. And that's what we'd anticipate. As I said before, iNKT cells sit on that innate killing mechanism spectrum as well. So great that it works quickly, but what we're more concerned with is long-term survival. So this is a similar type of experiment now where we look at a survival curve where we go out for much longer time periods. So if the animals were to all stay alive, it would be 100% alive up the top. If they were to die and they step down as they do so, then we get down to 0% of the animals alive. And as opposed to 3 days, we're out to 90 days here. So when we look at the black, which is the salt water, blue or green, which is the T and iNKT cells without any targeting, they all die between 40 to 60 days. When we look at the comparator, the CAR-T, as I said, that's similar to what Emily Whitehead would have taken. It's excellent data. 60% of the animals are alive at 90 days. It's just when we look at the iNKT cells, it's in the order of 95%. So we're seeing better survival, which we think is a result of that really robust upfront killing. And just one last piece of data, I think this is quite interesting and something we'd like to investigate further, and has given us some insights into additional indications that we might target. So these are 4 mice that actually took our therapy. I didn't say it on the previous slide, but we had 19 animals treated with the CAR-T or the CAR-iNKT. So 4 of the mice were treated with our therapy. And at the earliest time point for each here, so it's going from 10 to 52 or 17 to 59 for the second mouse. Early days, we see that they are looking cancer-free. We've eliminated most of those cancer cells. And then I think it's quite clear what you consider at the 20 to 30 day mark is there's solid tumor or tumor coming back in the brain. And that's the case for all 4. And so the thought process as you're collecting this data is that the animals will completely relapse and they will die. And the curve that I showed on the previous slide wouldn't look as appealing. And instead what happened, and this is with no further dosing with the iNKT cells, they actually underwent what we now termed a spontaneous secondary remission. So they were able to resolve the cancer a second time. Now, we believe that means the cells are able to last in the body, they're able to cross the blood-brain barrier, and they're able to have an effect on a more solid tumor-like cancer, which is important and exciting considering where we're taking the platform next. So where are we now in terms of the major activities for taking any product into clinic? We're very comfortable with the activity based on how quickly and swiftly these guys kill. We're very comfortable based on some other clinical data from other groups that the iNKT cells will be safe, and the CAR we're using has also been incorporated into approved products. So we're very comfortable with our safety profile. And as I said, we've actually announced it earlier in the year, we've received our GMP lentivirus, which is how we get the genetic material into the iNKT cells. So we're looking forward to scaling that up, obviously, and that's what enables us to make multiple doses per run. And then, of course, taking that into clinical trials in the second half of calendar year 2024, which is very exciting. Now, I'll just switch gears a little bit, but I'd like to talk about our strategies in place and our plans to take our iNKT cell platform into solid tumors. And many people probably know this, but when we think about the split between blood cancers and solid tumors, it's in the order of about 90% of any newly diagnosed cancer will be some form of solid tumor. So we see this as a really important societal issue, but of course, for the iNKT cell platform does present as a fairly large commercial opportunity as well. Now, in terms of thinking about a blood cancer and a solid tumor, they are quite different in how they present. And looking at a blood cancer, thinking about it as two cell types, let's say cancer cells and immune cells in solution, there's a reasonably good chance that they can find each other. And so the CAR-iNKT cells can kill the tumor cells. Now, when we think about solid tumors, I think it is quite a different beast that we're dealing with. And most people now synonymously know that as a lump. So that's what we see. And that lump is trying its best to grow without our immune system getting involved and breaking that down. So iNKT cells have a unique set of properties that we believe set them up well to work in solid tumors. And for patients that have presented in the past with things like head and neck cancer or colorectal cancer, having a low number of iNKT cells initially actually result in a much worse prognosis than for those patients that have high iNKT cell numbers. So in addition to that, we know that they also are capable of infiltrating inside tumors. And there's some cells in the tumor microenvironment called myeloid-derived suppressor cells or tumor-associated macrophages, which normally are our allies, but they turn rogue and start to support the tumors. And iNKT cells have been shown in preclinical models to help block or kill those types of immune cells that start supporting cancer cell growth. So what we're doing at the moment is we're working really with three major strategies to target solid tumors. So the first is we're working with different groups and scanning the landscape to find novel cancer targets that we can then utilize to have CARs that go into our CAR iNKT cell platform. And we licensed one of those in October last year from Sparks Group. Now we're also working to enhance the iNKT cell platform for solid tumors, and we licensed in what we call an armoring technology from the University of North Carolina only about 8 weeks ago. And we're working to create unique partnerships with groups where we see that there's a 1 + 1 = 3 approach, and we've got a number of discussions ongoing at the moment. So in terms of adding additional CARs or strategy one, as I said, this is something that we've already embarked upon. And the beauty of it is that I mentioned, and I went through our manufacturing pathway before, the only thing that we need to change when we think about having new CARs or new targets is actually just the lentivirus, which fits in at this stage, which is what we use to deliver the genetic material into the iNKT cells. The remainder, which is what we've been working hard on over the last sort of 18 months to 2 years, that stays the same, and naturally there'll be some level of optimization. But really just getting one new raw material is the major change, which is excellent. So when we think about certainly costs and speed to getting our pipeline products into clinic, we expect that to be a much faster pathway. And as I said, I'd just like to talk about the target that we did license last year. So it's called Claudin 18.2. The group that we licensed that from, Sparks Group, developed a monoclonal antibody, which is what we use to integrate into the iNKT cells. They had taken that all the way to an open IND with the FDA, so they'd already done the necessary safety specificity studies, including non-human primate studies. So we're very comfortable with that from a safety perspective. In terms of a target, Claudin 18.2 is found on gastric cancer, pancreatic cancer, and some ovarian cancers and some lung cancers as well. Now it's quite it's still an exciting target. I'd say arguably still somewhat new. Nevertheless, it is a validated target. So there's a large group called Astellas Pharma. They brought their monoclonal antibody from another company called Ganymed in 2016 for 422 million euros up front and 600+ in milestones. And they are expecting to get that approved in calendar year 2024. And predicting sales of 600 million to 1.3 billion. And that's off the back of 2 phase 3 studies that demonstrated extension of lifespan for these gastric cancer patients of 2.5 months. So we're quite excited by that, because having a Claudin 18.2 CAR iNKT cell program and a bar of 2.5 months extension to yield a blockbuster drug is quite a proposition. So certainly the gastric cancer market on its own is set to be quite a big one by 2030, and that doesn't also account for pancreatic cancer and the other Claudin 18.2 positive tumor types. And I just want to quickly touch on our armoring strategy. This is something that we had a handle on for some time, but we licensed that in about 8 weeks ago. And again, we think this is a really nice addition to our solid tumor programs. So it's termed IL-12TM, which I understand is a bit of a mouthful, but what that actually is, we call it a cytokine, and it gets inserted into the iNKT cells. And as I mentioned before, when we change a CAR, then we can add the IL-12TM in the same delivery vehicle for the genetic material. So we don't have to change our manufacturing process for that one either, which is excellent. And why we license that in, why we're excited by it, is because we believe it can enhance the iNKT cell platform, which we think is important for solid tumors. And what has been shown now already in animal models is that it helps for the iNKT cells to expand more and survive longer in an animal model setting. And when we look at animals after 4 weeks using this technology versus without, we see that there's 10 times more circulating CAR-iNKT cells. So just thinking about that in terms of an analogy, if you've got soldiers on a battlefield, having 10 times more of those cells in the bloodstream to attack the tumor cells is a good thing. Now, of course, then what we saw out of that is what we were expecting, but it does yield superior anti-tumor activity. And that's been done in a solid tumor model, which is a pretty nasty pediatric cancer called neuroblastoma. So that's now been published already in the prestigious journal Nature Communications, which was published about the 2nd of January early this year. And it's also, the work's been completed by, or in the lab of the gentleman that essentially created the first CAR-iNKT cells. So excellent to have a relationship with someone with that level of background in CAR-iNKT cell biology. So in terms of our pipeline, we are quite excited by where it sits today. As I said, we've got ALA-101, which we're developing for CD19 expressing cancers, lymphomas and leukemias, ALA-105, which is being developed for gastric and pancreatic cancers, as well as our IL-12-TM, which we're integrating into our solid tumor programs as well. Now, importantly, I'd just like to talk or touch on some of the milestones that we've got upcoming for 2024. So big goal for the year is to get our lead program, ALA-101, into clinic. So we're aiming to complete the manufacturing for phase 1 quite soon. And while we're doing that, we're completing the preparatory activities for reaching phase 1, including preparation of reg dossiers, engagement with clinical sites and KOLs. And of course, that would all proceed then getting our ducks in a row to commence phase 1 in 2024. For ALA-105, as I said, this is our gastric or pancreatic cancer targeting product. We're initiating proof of concept testing, which we expect to have in the first half of this year. And once we've got that in hand, we'd then optimize the CAR construct, so we've got the best efficacy possible, and that would proceed getting or using those constructs to generate animal data, and then formally commencing the manufacturing studies to take that one into clinic. And as I said, for our IL-12-TM, looking forward to integrating that into all of our solid tumor programs, and then testing that also in a range of anti-tumor models. And in addition to that, for that program, we're excited to enter into a sponsored research agreement with Professor Gianpietro Dotti, as I mentioned a moment ago, who was one of the pioneers of CAR-iNKT cell work. So exciting 12 to 24 months ahead. And I'd just like to summarize here and just say, as I said, we've got a very exciting novel allogeneic CAR-iNKT cell platform, which we think is a great way of getting off-the-shelf products developed for cancer treatment. We see that the improved manufacturing and logistics is incredibly important, given where we still sit today with the manufacturing challenges and the price tags of CAR-T. In addition to those things, we know that CAR-iNKT cells have properties that other immune cells, such as T cells and NK cells, don't have. We're excited to take our lead product, ALA-101, into clinic. We're also excited to continue expanding the pipeline through our things like ALA-105, which we're using for gastric cancer and pancreatic cancer treatment. And also for these reasons, we do see that we are in a good position as a company, and certainly looking forward to enjoying shareholder growth and value creation over the years to come. Thanks, Matt. I'll hand it over to you now to see if there's any questions.
Thanks very much for the presentation there, Michael. Once again, a reminder, if anyone has a question, please feel free to type it in within the function within Zoom. Michael, the first question just pertaining to the announcement today was just asking for if you had any comments around the collaboration with Imugene ceasing.
Yeah, look, I'll say what I can. Imugene have decided that the partnership is not a strategic priority for them, but I'd just like to make it clear that the termination of that has no bearing on ALA-101, and certainly over the last 18 months since we started that agreement, we've licensed into, I think, really amazing technologies in Claudin 18.2 and the IL-12-TM, which will integrate together. So certainly looking forward to getting those lead product into clinic, and those products coming up pretty fast behind to tackle solid tumors as well.
Very good. Just in terms of assets, are you comfortable with where you're at, or would you look at adding more to the pipeline?
Yeah, look, I think it's a good question, and something I continuously do is survey the landscape for other products that are under development or looking for products where there could genuinely be a synergistic outcome for combination. So we are in discussion with a number of groups for where we think there are synergistic programs that could be developed together. And in addition to that, we also look for new cars where we think we've got shots on goal and can expand the utility of the program. So again, I think it's always managing what we've got on our plate currently, but making sure that we're also continuing to be the leader in iNKT cell biology.
And I've heard the comment from one of your major shareholders a number of times that your asset is really one of a kind in the world. Can you just sort of discuss the significance of that?
Yeah, absolutely. So I showed a slide a moment ago that demonstrated how many players there are in the iNKT cell space, and it is such a few, right? And I think that's really important when we think about all the properties that I've raised about iNKT cells in terms of the fact that they can be used off the shelf, the fact that they infiltrate tumors, they do influence the tumor microenvironment. We see all these things as really important in creating the best possible therapeutics that we think can have an impact in the lives of cancer patients. And that's our goal and always will be. So to be one of a handful globally working on this type of approach, I think is sensational. And except for Appia, who are also working on CAR-iNKT cell products, the others are really still focusing on unengineered iNKT cell products. And I think they're working to bring CAR-iNKT cells up. But I think it certainly sets Arovella up to be one of the leaders in CAR-iNKT cell biology, which is excellent.
And obviously working toward the clinic soon, which I know everyone's excited about. But again, my question is, how should investors, I guess, sort of monitor or judge your progress in the lead up to that, and also once your phase one's underway?
Yeah, good question. What we intend to do, obviously, is provide updates throughout the year, some manufacturing updates, aiming for that to be shortly, as well as any progress with regulators. We would announce that as well. Obviously, if the trial's given the all clear to start from regulators, that would be a key indication, and I think a pretty important step for the company. And then things like dosing of patients and so forth. So because this is going to be a trial that's not blinded or randomized, we do envisage that we'll be able to present data. Now, we'll be managing how we do that so that it's done in an appropriate manner. But it doesn't mean that once we start the clinical trial that our investors are having to wait for 2 years in order for them to get a readout or to get an understanding of how the trial is progressing.
Excellent. Another question that's come through is, you mentioned the cost of traditional production of a patient's cell for reintroduction to their system, and that your process of taking blood from a healthy person and growing the numbers to reintroduce is much less expensive. Are you able to give any sense on how much less expensive that process might be, as this will give us a sense of competitive advantage that your process might deliver?
Yeah, absolutely. And look, we don't have the exact number right now, but what we can say is, because we're expecting to have hundreds and hoping to push into the thousands of doses per batch, it's going to be a very small fraction, I think, of the current treatment costs. But until we have a firm handle on a few things, one being the dose level, that impacts obviously how many doses we end up getting out of every manufacturing run. We can't conclusively say what that number will be. And of course, that's a number that also gets determined while we're doing our clinical trials, our dose escalation, and into phase 2 as well.
Great. Well, thanks very much for the presentation and taking those questions, Michael. It's much appreciated.
My pleasure, Matt. And I'd just like to say thanks again for putting the event on, and thank you to everybody for taking the time to dial in. Appreciate it.
Thanks, Michael. And yeah, that actually wraps up today's sessions for the conference. We've got another day tomorrow with about another 10 presentations. All the details are on the website. You can access it just via the NWR Communications website. Thanks to all the speakers today, to BioShares, who are a supporter of this as well. And for anyone who missed any of the earlier sessions who might be interested, they're all accessible. The replays go up shortly after the live session concludes, so please feel free to jump on and have a look if there's anything that you missed that you might find interesting. But we'll leave it at that for now and see everyone tomorrow.
Cheers. Thanks, Matt. Bye.
Thanks.